So I am an assistant professor at the International Laboratory for Human Genome Research of UNAM in Mexico. So I just returned to Mexico in 2021 to start my own group and do some research on genetic disorders and genomic disorders. So I'm really happy to share today with this audience some of the work that we're doing more on the bioinformatics side, doing genome analysis of repeated sequences. So most of you probably have seen this statement that any human is 99.9% identical to another human. However, this is somewhat inaccurate because the reality is that two humans are 99.5% identical to each other. And this difference comes from not just from SNPs, which is the 99.9 number, but also from other types of variation that are usually not considered when we're talking about the similarity between humans. So as I was saying, the 99.9% number comes from just single nucleotide polymorphisms, which are regions where just one nucleotide is changing between two individuals, like in this case. But there's a lot more other variation that it's, again, generally not considering this number, and that makes this other 0.4% of differences between humans, and that is structural variation. And there are different types of structural variation, and I'll talk a little bit more about some of these different types, but more generally, so there can be deletions, which are losses of segments of DNA, and this can be from a few base pairs to and from small indels to very large segments, some of them several kilobases or even megabase size regions. There can also be insertions, translocations, inversions, duplications, replications, and different copy number changes. And so back in 2004, the recognition of these amount of the structural variation that accounts for variability between individuals and also in different populations, because some of these structural variants are also population specific, was what prompted for this publication in Science to call structural variation and human genetic variation in general the breakthrough of the year. So this recognition that humans were a bit more different than the initial number that we got just from looking at SNPs. So as I was saying, some of these changes involve duplications, replications, and different and deletions as well. So different changes in the number of copies of certain regions. So generally, copy number variants are these regions in the genome that deviate from the expected diploid copy number state. So either less than 2 or more than 2. So these can be either deletions, like in this case, or duplications more commonly. But again, we also can have triplications and other further amplifications of more than 2. These are important because, again, many of these will be in non-coding regions, so they won't encompass genes, and many of these are polymorphic within individuals and then among different populations. But some of them, when they encompass dosage-sensitive genes, they can result in disease, and these are usually called genomic disorders. And so these genomic disorders are the consequence of DNA rearrangements that involve relatively large regions of the genome, and again can change the expected diploid copy number of that region and the genes involved in it, or also the order. So some of them will be inversions that changes the architecture of the region, and these can cause disease through different mechanisms that I will talk about. But basically, these are, we call them genomic disorders because they involve genomic regions. It's different from genetic disorders that are usually referred as monogenic conditions, usually caused by alterations in one specific gene or so. So this is a non-comprehensive but illustrative list of some of the more common genomic disorders. And these genomic disorders started to be recognized in the 1990s, mainly through the utilization of fluorescence in situ hybridization that allowed to see that some regions were changing in copy number. Obviously, before that, also big changes in chromosomes through karyotyping also prompted people to start looking at these large structural changes in the genome. And then as we have improved the resolution of different technologies, we have been able to identify even more of these genomic disorders. But one thing that I want to draw your attention to is that some of these say recurrent, which means that the rearrangements, the same rearrangements can occur multiple times in different non-related individuals. So some of these are recurrent, some we've also seen non-recurrent rearrangements, but also some of them you can see here that they have deletion, duplication, deletion, duplication, deletion, duplication, and they are recurrent rearrangements. And what this basically means is that some of these rearrangements, like the example of Charcot-Marie-Tooth disease type 1A, which is one of the very first genomic disorders to be identified and for which the genetic cause was described. So in this case, this is a very large duplication that you can see here that encompasses this dosage sensitive gene called PMP22, protein myelin, peripheral myelin protein 22. And so as you can see here, so this is a picture of an array CGH, array comparative genomic hybridization assay, that shows that here there's an increased copy number for this region, whereas here there is a reduced copy number for this region. And this is, again, so the duplication causes Charcot-Marie-Tooth type 1A disease, whereas the deletion causes a disorder called HNPP, which is hereditary neuropathy with liability to pressure pulses. So these two disorders are mirror disorders in that, again, one is caused by the duplication, one is caused by the deletion, and it's exactly the same region, but these rearrangements are mediated by what we call low copy repeats, or regions that are flanking these repeated regions that are flanking these intermediate region that, given their specific characteristics that I will go a little bit more into, can cause these genomic rearrangements in either the duplication direction or the deletion direction. And this is why we have these reciprocal genomic disorders caused by either deletion or duplication, or even sometimes amplification of the region because of these repeated regions that are flanking these regions of interest. So why is this important, and how do these rearrangements affect human health and disease? Well, they can cause disease by different mechanisms. So the most common one is by dosage sensitivity. And I mentioned before that, so if some genes, like in the case of PMP22 here, are involved in the region that's changing its copy number and involved in the rearrangement, and this gene is important to have 2 copies of the gene to have normal cellular function, and we lose one due to a deletion, then that will cause a disease. Similarly, if we increase the number of these dosage-sensitive gene that can also cause disease, because the amount of this protein is very tightly regulated, and important that it's that it's in 2 copies rather than more or less copies. And again, if we have increased number, like triplication of the region, that can cause disease, and generally this is more severe disease. So we have seen some cases of triplication of the CMT1A region that is associated with much more severe and earlier presentations of CMT. Another way in which these genomic rearrangements can cause disease is through deletion of different and multiple genes. So as you can see here in this diagram, so PMP22, which is located right here, it's not the only gene in the region, so there are different genes. We do know that this is the most important gene, that it's the dosage sensitive one, that it's responsible for CMT and HMPP, because we also have seen patients that have mutations in the gene. And if it's a loss of function, then you will have HMPP. And if it's a different type of mutation, you can have CMT1A. But there are other genes involved in this rearrangement. So in some cases, the phenotype that we see in some patients can be a product, the result of multiple genes that are being altered in their copy number or lost through deletion, and not necessarily just one gene that's dosage sensitive. Additionally, if some of these rearrangements occur in the middle of the gene, they can also disrupt the gene and basically causing a knockout for this gene in one of the copies, and then having haploinsufficiency for the gene. In some other cases, we have seen also fusion of these of genomic regions due to a rearrangement. In this case, if we have a deletion in this region, then we can have a fusion of these 2 genes. And depending on the product of these fusions, sometimes these can drive certain diseases. And we think that this is especially relevant for some cancers. Additionally, I mentioned before, another mechanism by which these rearrangements can cause disease is through positional effects. And what that means is that in some cases, the genes remain intact, but what is being affected is regulatory elements. So either the loss of some regulatory elements, or the if we have, for example, duplications or other type of amplifications, we can have a disruption of the distance between the regulatory element and the gene. And then this affects how the enhancer or the suppressor is actually regulating the expression of the target gene, because it's too far from its target, and then we lose that specific regulation. Or again, if we have a deletion, we can lose the specific element and lose that regulation as well. So again, there can be different mechanisms by which these rearrangements can impact gene expression and gene function, and consequently show as a genomic disorder. So one way to understand these rearrangements is to look, well, first is at deletions and rearrangements in human patients, but also look at the what we call the breakpoint region, which is this area where the union, right? So if there was a deletion, like in this case, where is this break happening, and what is happening in this sequence? And by looking at these breakpoints, we can start to identify some patterns and some characteristics of these genomic rearrangements. So in this case, for example, you can see the sequence of the proximal region here. This is a paper I worked on very early on during my PhD, but I thought it exemplifies really nicely sort of the analysis of breakpoints, or in this case, deletions in the GJB1 gene. that cause another type of Charcot-Marie-Tooth disease that it's X-linked. So in this case, you can see the proximal region here and the distal region to the deletion, and then the shared sequence in the middle. So this small track is shared between both the proximal and the distal region. And then some other cases, we can see these, what we call these blunt cuts in the DNA, where we really don't see any more sharing, as you can see here in the proximal versus the distal sequence. We don't see any more sharing, and sometimes we have some addition of these, a few bases here in the repaired breakpoint. And these two are different mechanisms, indicate to us that different mechanisms produce these rearrangements. And so through this analysis of different breakpoints of deletions and duplications found in human patients, so we can start to understand some of the mechanisms involved in the generation of these rearrangements. So there are three main broad mechanisms that can cause genomic rearrangements. So non-homologous end joining is the mechanism of repair that the cells use when double-strand breaks occur in the DNA, and then they are preparing to replicate their DNA. They need to repair things, and then basically they take these two ends and try to paste them together, basically. And this repair mechanism, usually, again, because we have these double strand breaks, so we have these repairs, and sometimes we have the addition of a few base pairs. But basically, this breakpoint that I showed you here, this is a very nice example of a breakpoint that we would see that was mediated by non-homologous end joining. And again, so these can happen through exposure, for example, to X-rays, which cause double strand breaks. And again, if the cell is in an emergency state trying to repair the DNA, it will resort to non-homologous end joining for this. And there's really not much that we can do to predict where these cuts will happen, because again, these cuts will be randomly happening based on exposure to generally ionizing radiation. So they can be kind of random and not much that we can do there. However, for the other two mechanisms, non-allelic homologous recombination, or NAHR, and more recently, fork styling and template switching and microhomology-mediated break-induced replication, which I will explain, so you don't need to figure out anything from the names. But for these two mechanisms, we can actually understand what is happening and how we can leverage this understanding to do predictions. So for non-allelic homologous recombination, this is based on large tracts of highly identical repeated sequence that can be either in direct orientation, meaning both repeated sequences are on the same strand, or in inverse orientation, where they are in different strands. And then, so the cell will use these repeated sequences to during DNA replication to similar to homologous recombination that happens to produce recombinants normally. In this case, when the cell is replicating its DNA, it will find these repeats, and then sometimes the fork, the replication fork, will separate and then join again, and then try to pick up where it left the replication strand. And sometimes it can make mistakes. So it can then, if it was replicating through these black repeat, then it finds something that looks very similar to this and take this other repeat that it's in the same orientation and continue replicating. But that means that this intermediate region will be lost because, again, it skipped this part. And so this is how we would get a deletion. Similarly, if it's going through this repeat region here and then it skips, it can jump to this white one and then sort of skip again and come back here. And then that can cause a duplication or a further amplification of the intermediate region between this black and white repeat. So similarly, although in the opposite direction. So if the polymerase is replicating through this region and then it encounters a similar region, but it's on the other strand, so then we can have this alignment of the repeated regions that will cause an inversion of the intermediate region. And so this is how very similar, highly identical regions that are distributed around the human genome can cause all of these types of rearrangements. And this is also why, because we have these repeated regions in very specific sections of the genome, we can have recurrence of some of these rearrangements because they will be substrates that are really recombinogenic through NAHR mechanisms. And so we can have non-related patients that will have the same rearrangement over and over again through this NAHR mechanism. More studies of other types of rearrangements, which generally are considered non-recurrent. So again, looking at different deletions and duplications and inversions of patients with genomic disorders, we started to see that not all of these rearrangements were happening using these same repeats that are used for NAHR. And then some of the rearrangements had different breakpoints and were occurring at different locations. And again, through the analysis of the breakpoint sequence of these rearrangements, we started to see that there were other mechanisms that could also use repeated sequences, but much smaller and more divergent. So less than 90% identity between the sequences. And this is what was called microhomology, because the homology is smaller. So microhomology mediated break-induced replication and fork-styling template switching. And basically what this means is that while there is replication happening, again, the replication fork will stall. And then this replication fork will, in some cases, jump to homologous regions that are close by that can be used to continue priming the synthesis. But then they can jump again, and then in these very complex regions with multiple repeats, and this can cause these more complex rearrangements where we see contributions from different repeats that share microhomology regions. So again, this analysis of breakpoints really had shed light on the characteristics of these repeated sequences that can serve as substrates for the occurrence of genomic rearrangements. So some of the characteristics that we know are important, as I mentioned before, orientation, whether these are in the same or in the opposite strands. The size. So again, larger repeats will be better substrates for NAHR versus MMBIR and FOSTAs require much smaller repeats with much more divergence that can be utilized in these mechanisms, whereas for NAHR, high identity is usually required for the recombination proteins to recognize these repeats as substrates. Also, the distance is important. So if these repeats are too far away, then the likelihood of them recombining through NAHR is much smaller. So if there are also clusters in different complex regions, they are more likely to have rearrangements as well through MMBIR. And also that allows us to look at density, again, some more complex regions that are very repeat rich, and the distribution genome-wide of these repeated sequences. Again, since 2004, where we started to recognize really the amount of structural variation, and again, previous work on genomic disorders and the recognition of these low copy repeats that contributed to NAHR and recurrence of these rearrangements in different patients. So we started to look at again, different characteristics in the distribution of repeats across the human genome, and how leveraging this knowledge of these characteristics of repeated sequences can help us predict where some of these rearrangements might occur. So this is from a paper we published back in 2010, where we were looking at the fraction of the genome that was completely identical, 100% identical. And again, some of these 100% identical regions are embedded within much more divergent regions, so you can have a drop in the identity as you expand the repeat, which will be important for the work that we have done more recently. But we wanted to see how much of it is highly identical, and then what was the percentage of the genome that was of this type. So we determined that 2% of the genome is identically repeated. And again, you might think like, well, 2% is not too much, right? But let's just remember that the coding fraction of the genome, right, the parts of the genome that are making proteins is also 2%. So it's pretty significant in terms of potential impact, again, for rearrangements and another type of variation. Some of these highly identical regions obviously will overlap with known repeated elements like lines and signs, transposons, ancient transposons, satellite DNA, and other microsatellite sequence and other repeated sequences, but some of them are actually in genes. So here are two examples of highly identical repeats that are like right in the middle of, in this case, 3 different genes, and in the first 2 cases involving actual exons. And in the last one, these are in the intron of this RNF13 gene. But again, some of these repeated sequences can actually be in the middle of genes, and in this case, for example, can cause fusion genes, and in this case, we were looking at inversions, so they can cause fusion of these transcripts, and again, this variability in this gene expression. So again, in this study, we were looking at highly repeated sequences, more than 95% identity, because we were looking specifically for NAHR. Again, this was around 2007, 2010, when the mechanism that we were most aware of that was driving genomic rearrangements was NAHR. So we were looking at these very highly identical regions. In order to predict genes and parts of the genome that could suffer rearrangements. We also looked at inversions. So deletions or duplications are generally better studied because they are more obvious. They are easier to detect because you see either increase or decrease copy number. However, inversions are less characterized because they are copy number neutral. So you won't see in an array CGH or a FISH really much of a difference in a patient that has an inversion. Because you are not actually changing the copy number. However, as I mentioned, some of these inversions can truncate the gene or cause disease through fusion genes or through positional effects with regulatory elements. So back in 2013, we looked at also substrates for inversions in the human genome, looking again at regions of more than 1 kilobase and 95% identity or more in inverse orientation that could actually mediate inversions and possible genomic disorders through inversions. So again, this is just the map of that analysis. And as I mentioned before, as we continued to study these rearrangements, so we realized that some of these non-recurrent, more complex genomic rearrangements were actually being caused by a different mechanism that didn't require these long stretches of highly repetitive sequence that it's required for NAHR, and that was how FOSTES and MMBIR were sort of identified and characterized as mechanisms driving these other type of rearrangements. So again, here you can see some examples even in the same region of CMT1A. So here you can see the 1.4 megabase region that is usually deleted or duplicated in HMPP or CMT1A respectively. But then here we have other patients with CMT that have, in this case, deletion, in this case is duplication, encompassing PMP22 as a dosage-sensitive gene, but also very different from the recurrent rearrangement. And then looking at the breakpoints, we start seeing also some that are more complex, which involve not just one event of deletion and duplication, but also combinations of deletion, duplication, and even inversion. So in this case, duplication, triplication, inversion, duplication rearrangements. So again, very, very complex rearrangements. And again, through the analysis of these breakpoints, that's how we realize that these smaller fragments that are more divergent can serve as substrates for this type of microhomology mediated rearrangements. So more recently, as you are probably aware, we also had the completion of a complete human genome through the telomere to telomere consortium and the sequencing and assembly of the T2TCHM13 genome, where additional sequence was found, including 51 megabases of segmental duplications, which are a specifically defined type of repeated sequences, more than 1 kb in length and more than 90% in identity, which overlap a lot with the low copy repeats that we see for a lot of the genomic disorders. So overall, based on the now complete assembly of the T2T genome, so the authors reported that about 7% of the human genome consists of segmental duplications. And one important contribution of the T2T genome was also that now we have sequence for the acrocentric chromosomes, which we didn't have for the reference genome. And as you can see here, so you can see for chromosome 22, 21, we see these red lines here that indicate previously unresolved in the reference genome segmental duplications. Also here, chromosome 14, 15, 13. So a lot of resolution that wasn't there, that we didn't have before for the reference genome, was added by the T2T genome, and 45 to 68 megabases was added as part of the acrocentric short arms, and many of that is repeated sequence. So with that new knowledge, new assemblies and that, so we set out to ask, well, now that we understand some other mechanisms for genomic rearrangements, and we have these other alternate assemblies, what is the contribution of repeats in the human genome to making to creating substrates for genomic rearrangements and different mechanisms for genome rearrangement. So this work is some of the stuff that we have been working over the last 2 years in my lab here at UNAM. And this work has been led by Luis Fernandez Luna, who's an undergraduate student from the Genomic Sciences program. And some of the initial work was started by Carlos Aguilar Perez, who is now a PhD student at the Max Planck Institute in Germany. And what we've basically been doing is using 3 different human genome assemblies. So the GRCH37 or the HE19 reference assembly, the newest one with more gap closures and increased resolution, the GRCH38 assembly, and the T2T CHM13 assembly from the telomere to telomere consortium. And the rationale for using the 3 assemblies is that most of the previous work on doing these maps of repeated elements had been done on the HE19 reference, but we hadn't updated that work for HE38, which is the latest reference, and now again having the T2T genome. So we were also curious to see how these the reference genome and the new T2T genome compare in terms of repeats and regions that might be at risk for rearrangements. So we also used updated parameters. So again, most of the analysis before had been done using parameters that were very tailored towards identifying substrates for non-allelic homologous recombination. So now we updated our parameters considering experimental observations that we had over the years. We and others had seen for rearrangements in human patients, and the occurrence of complex and non-recurrent rearrangements. So we updated our parameters to a minimum length of 200 base pairs, which appears to be the minimum length of a repeat that's needed for the mechanisms that either, in this case, MMBIR or FOSTAS, to recognize these as a microhomology substrate, and also an identity of 80%, which again is also based on experimental observations, what we think is the minimum identity required for this identification of microhomologous regions that can serve as substrates for rearrangements. So we used the LASSI algorithm to perform chromosome-wide pairwise alignments to identify repeated pairs across the different chromosomes. And because of the way LASSI is coded, so sometimes the alignment might fall off, even though closer by there might be another chunk of the repeat. So we developed an algorithm to collapse overlapping or very close nearby repeats that can be substrates for microhomology mediated rearrangements in complex regions, but ensuring that these collapsed repeat regions still had at least 80% of identity between pairs. So these are the general metrics of our both direct and inverted repeats that we detected through this analysis. So the median length for direct repeats is about 500 to 525 for the T2T genome. Similar numbers for the inverted repeats from 469 to 520. The mean length it's a bit higher. And then again, the median identity is also around 84% and 85 mean identity for both direct and inverted repeats. We also looked at the repeat content for these detected repeats. So again, we knew that many of them are overlapping with known repeat elements such as signs, which again we see here much more represented. And again, most of these regions had been excluded previously because we were looking for NAHR substrates, but we've come to realize in the years after that ALU recombination plays a huge role in some of these rearrangements. And so ALUs are a class of signs that depending, and some of them can be very divergent, but there are some others that are still pretty similar and can be extremely important substrates for rearrangements, including some that are in exons in coding genes. So we also see, again, as expected, segmental duplications, satellite DNA, LTRs, lines, and other types of repeated elements. And again, the distribution for the 3 different assemblies is very similar. We do see an increased detection of repeats in the T2T-CHM13 assembly, which is, again, not surprising because a lot of these 8% additional sequence that was added to this assembly compared to the reference genome, it's a lot of that is repeated sequence. So we do see that in our analysis. This is the genome-wide and per chromosome distribution of the direct repeats. And again, we see distribution of repeats across all chromosomes. And just to point out that, again, the distributions are very similar for most of the chromosomes, with the slightly higher numbers for T2T, but where we see much of the difference, it's precisely in the acrocentric chromosomes, as I mentioned before, that weren't resolved in the reference assembly, and now we have base pair resolution in T2T. So we can see here this increased number of direct repeats, and you can see here also in the HE19 and the GRCH38 assemblies, we don't have data for the acrocentric chromosomes, whereas for the T2T chromosomes now we have repeat elements identified there. Similarly, for the inverted repeats, we see the same trends, similar distribution for the 3 assemblies, but slightly higher for T2T with an increased representation of repeats in the acrocentric chromosomes given the new sequence. We also looked at, well, how genes are impacted by these repeats and what genes are at risk of undergoing genomic rearrangements or structural variation in affected by these repeats. So about 900 protein coding genes overlapped with direct repeats and about 800 with inverted repeats across the 3 assemblies. And we did a gene ontology enrichment analysis to see like whether these genes were overrepresented for certain gene ontology categories. And again, not too surprising and reassuring, because we have seen these before in previous analysis of repeated sequences of segmental duplications and such, that there is an enrichment for olfactory receptors. We know olfactory receptor clusters in different chromosomes are very copy number variable. So a lot of these repeats are mediating these constant rearrangement in different individuals and populations of olfactory receptor clusters. And also, again, so we see enrichment for gene ontology terms associated with olfactory receptors, and also for immune and environmental sensing functions. So again, we also know that a lot of the immune genes can be very variable, which also that gives some advantages to diversify the repertoire for these genes to interact with the environment. So that's something that has been documented before. So in summary, and I just want to leave some time for any questions that there may be, but in summary, structural variation contributes to more variable base pairs in the human genome than SNPs. We know that the human genome is highly repetitive. Between 30 to 40% of the sequence is composed of repeats. Some repeated sequences can serve as substrates for different mechanisms to generate genomic rearrangements. And when these rearrangements encompass genes, especially dosage-sensitive genes, this can cause genomic disorders that we can see in human patients due to dosage or positional effects. Studying the characteristics of the repeated sequences and of these genomic rearrangements to understand the repeated sequences that are acting as substrates is important, so that we can also make predictions, make this type of genome-wide analysis to predict what regions may rearrange in some other patients. And these type of genome-wide analysis are good catalogs of repeated sequences that can inform the location and distribution of unstable regions in the genome. And these regions, although can rearrange and cause disease, as in genomic disorders, they can also contribute to human polymorphic variation and variability in creating variation to enhance sensory and immune adaptation, and ultimately genome evolution is part of why these regions are still part of our human genome and contributing to creating variation. So with that, I just want to again acknowledge the students that are working on these projects in my lab. I mostly talked about the work that Luis here has been doing, Carlos, who was previously in the lab, but also many other students working on different projects. Also collaborators working with us on this project, Claudia Carvalho-Fonseca at the Pacific Northwest Research Institute, Christopher Grachowski from Baylor College of Medicine, and other collaborators. Special thanks to the National Visualization Lab that has the high compute cluster that we use a lot for our large-scale genomic analysis, and to the different funders that help us do this analysis. And with that, I'll be happy to take any questions from the audience.
Excellent, Claudia. It's a very interesting talk. So we do actually have a question already. It came about fairly early on in your presentation. The question was, can genomic rearrangements, quote unquote, cure disease the same way that they can cause them? Of the two, which is the more likely outcome?
Well, that's an interesting question because, again, technically, especially for the rearrangements that are mediated by NAHR, so these can be very dynamic regions and can rearrange very often. So, I mean, we know of examples, for example, in Drosophila, where some of the initial genomic rearrangements in eukaryotic organisms were observed in Drosophila through these amplifications in these chromosomes that made these very long fat chromosomes. And we know that some of those rearrangements can revert. So we see revertance for Drosophila, and that with humans, I mean, technically that's possible, but I mean, we often don't see those cases because I mean, people don't come to the doctor, right? We always have in medical genetics a bias to see people that have diseases, not people that sort of were cured from a disease or something. So, I mean, you could potentially have, I don't know, a parent with a CMT1A duplication, where maybe in the germline the duplication sort of reverted and one of the copies was lost. And then you have a child that doesn't have the disease, but we wouldn't see that that child because they wouldn't have clinically the disease. So again, technically, because of the dynamics of these regions, it can be possible that we have revertance for the rearrangements, but we don't see them. We mostly see the ones that cause disease.
That makes sense. So we do have another question. In the earlier study, 2% of the genome had identical repeats. What was the percentage in the current analysis?
So the first the one that had the 2%, so that was 100% identical. So 2% of the genome is 100% identical. In the current analysis, I don't remember off the top of my head right now what was the overall percentage that is involved. So we have the number of repeats and the overall base pairs, but I don't remember the percentage at the moment. But again, these are slightly different analysis in that the parameters for the new study involve smaller repeats with higher divergence, so at least 80% identical. So they are not exactly comparable. Of course, the latest one will be higher the percentage because we're looking at much more divergent sequences across the genome.
Okay. So my own background is studied a lot of introgression, but I used plants as a model. Now you said that there's when you're looking at humans, there's a limited threshold of similarity that you have to have. Um, you pointed out originally it was 1,000, and now you've reduced it down to 200 with that 80% threshold. In looking in plants, and this could just be plants being weird because it's what they do. There are some instances where you can get like a snip in the middle of a tandem repeat that will generate not necessarily an immunity, but a reduction in the disease phenotype. Um, have you seen anything like that sensitive in humans where something really small that shouldn't fit with all the other data? Because as you say, you do need a certain level of similarity before this reorganization really matches. Have you seen something that fine a scale in what you've looked at?
Not that I'm aware. I mean, so again, these parameters that we went with for this latest analysis are mostly based on observations, experimental work that has been done on hundreds of patients with non-recurrent rearrangements and looking at the breakpoints. But again, we do have a bias, right? Because
Mhm.
we wouldn't see, for example, cases of repeated sequences that didn't occur, the rearrangement didn't occur because there was like one change, and that prevented the machinery from recognizing that as a highly homologous region to recombine. So, I mean, I think those questions are really interested in terms of the mechanistic part of how these rearrangements occur, but those usually I mean, are better addressed on like doing actual experiments generally on bacteria or yeast, sort of looking at mutagenesis of repeated elements and looking at how much or less they recombine based on those changes, because again we wouldn't see some of those cases in patients.
Gotcha. Excuse me. Gotcha. That does make sense. So then, kind of a tangent question here. Of everything that you'd found while you were putting this together, what was in your opinion the most interesting result?
Well, again, I think so. This analysis that we did was partly because some like we hadn't updated some of the analysis from HE19 to GRCH38, and then we had the T2T. So I think some of the interesting stuff is really looking at the distribution. So obviously both the reference assembly and the CHM13 new assembly are not that dissimilar. The CHM13 is mostly European ancestry, and we see a similar distribution genome-wide of the repeats. I think having this new sequence in the acrocentric chromosomes is really interesting to start looking at some of these repeats as substrates for, I mean, we know a lot of the acrocentric chromosomes can cause translocations and fusions with some other acrocentric chromosomes, mostly because of the RNA repeat arrays that are there that, again, can serve as substrates for these type of recombination and rearrangements. So I think one of the future things that we are really excited about is also looking at other genomes, as we are starting to have more genomes, and see if there are differences in the distribution of repeated elements and regions that might be risk regions, and what genes are involved in that in different populations. So we're very excited to see at some point the new genomes from the Pangenome project and do this type of analysis there and have this broader view of the genome-wide distribution in different ancestries in different individuals for repeated elements.